Good afternoon, everyone, and thank you for tuning in. My name is Rishi Bansal, and I'm in my third year of undergrad at McMaster University. I'm a new CDTRP trainee as of last month, and I'm working with Dr. Maureen Mead on a project to survey the content, quality, and completeness of transplant recipient databases at transplant programs across Canada. It's my pleasure to be introducing Dr. Bethany Foster to you today, who will be leading this presentation. Dr. Foster is a director of the Division of Pediatric Nephrology at the Montreal Children's Hospital, which is a clinical epidemiologist and a professor of pediatrics at McGill University. Her primary research interest is in the long-term outcomes of children and young adults with kidney transplants. Dr. Foster has been funded by the CIHR and NIH to study immunosuppressive medication adherence and graft outcomes in adolescent and young adult kidney transplant recipients, whom she has identified to be at particularly high risk of graft loss. She has also highlighted important differences in renal allograft outcomes by recipient sex, the magnitude and direction of which vary by recipient age and by donor sex. So at this point, I'd like to hand it over to Dr. Foster. Please take it away.
Thank you very much. Okay, so I've been asked to speak to you today about sex and gender in transplantation research, and I understand that this is at least the second webinar you've had on this topic. I know Dr. Melk recently gave a webinar on a similar topic, and so I've tried to not overlap too much with her presentation. So the objectives of today's talk are first to consider the importance of both donor and recipient sex in transplantation, and I'm going to do that using a specific example. Well, a couple of specific examples, I guess. And also I will spend a little bit of time talking about the importance of recipient gender in transplantation, although not so much on that topic today. So just to remind everybody the difference between sex and gender, I'm going to first focus on sex. So sex is defined as the biologic attributes in humans and animals that are primarily associated with physical and physiologic features. So this is things like chromosomes, gene expression, hormone levels, etc. So this is really the biological part. So first, thinking about donor sex, anytime you're considering research, either fundamental research or clinical research in transplantation that has to do with the donor, it's important to consider the sex of the donor. So as you know, but perhaps isn't always reflected in the basic science literature, every cell does have a sex. And when you think about a donor organ, I've got kidneys here, but of course, you know, this would apply to any organ, liver, heart, pancreas, whatever. That organ is composed of cells that have a sex. So there could be an XX donor or an XY donor. And why is that important? Well, for a few reasons. There are obviously differences in the proteins expressed on XX and XY organs, the most obvious of which is the HY antigen, which is expressed on all male cells, and also, and not expressed on female cells. And there is no sort of analogous antigen that's expressed on female tissues that isn't expressed on male tissues. So it's just the HY antigen on male tissues. There are also sex hormone receptors on female and male tissues, and the density and expression of those receptors may be different in female tissues compared with male tissues. It's also important to think about the milieu that donor organ is in before organ recovery, because the milieu that it's in, it may differ by sex depending on the age of the individual. So a lot of sex differences are age dependent. Obviously in very small children, the sex differences may not be so large, but in young adults, there will be fairly large sex differences, and then postmenopausally, the sex differences may be less stark, although certainly still present. So in a female donor before organ recovery, there was probably going to be a fair amount of estrogen floating around. Not so in a male donor, where there's more testosterone in the bloodstream of the donor prior to organ recovery. So why is that important? A few years ago, there was this very interesting study done in mice, which showed that there are important sex differences in the tolerance of kidneys to ischemia reperfusion injury. So the first panel here shows the differences in mice tolerating warm ischemia. So this is what they did was ligate the artery, the renal arteries of male and female mice for different lengths of time, and they found that females can tolerate 28 minutes of warm ischemia compared with only 15 minutes in males. And interestingly, they were able to demonstrate that these differences in tolerance for ischemia reperfusion injury were mediated by the hormonal environment. So in panel B, you're seeing in the top two plots here, the triangles and the squares are neutered animals. So neutered females in the triangles, neutered males. They had fairly similar tolerance to ischemia reperfusion injury, and neither of them was as good as intact females. I haven't shown all of the figures here, but they did some nice experiments as well with knockout of the estrogen receptor, and found that it was the estrogen receptor and the presence of estrogen, obviously, that were necessary for this protective effect to happen. So it wasn't associated with donor sex. That's what's shown in panel C here. Where when the recipient was, so when they did transplantations, when the recipient was female, that's the circles and the squares, the two on the bottom, there was better tolerance to ischemia reperfusion injury than when the recipient was male, regardless of the donor sex. This last panel is showing differences in fibrosis on histology, where the female recipient, in whether the donor is female or male, did better than the male recipient with respect to tolerance for ischemia reperfusion injury. They also did some experiments showing that supplemental estrogen was beneficial in female mice and in neutered males, but not in unneutered males. And that was part of their line of investigation, showing that not only is estrogen protective, but testosterone may actually potentiate the injury, ischemia reperfusion injury. So this is a schematic just trying to summarize some of the differences that one might expect based on sex, both in the setting of a donor and the setting of recipient. And we're going to focus right now on differences by donor sex. So anything above this black line in the middle suggests that there's better survival, and down here is worse survival. So, for example, there may be intrinsic differences in organs based on sex. This is certainly the case in kidney, where we know that males tend to have a greater number of nephrons and larger nephrons than females, and so a male donor may actually provide a graft survival advantage over a female donor. However, as we just saw from the previous slides, females tend to have better tolerance of warm ischemia before the donor surgery, which is mediated by estrogen, whereas testosterone may actually augment injury. This is probably most important in the setting of DCD organs, donation after cardiac death, where the period of warm ischemia will be longer than in a neurological death donor. But it's important to consider in donor management, particularly in DCD, but also in neurological death as well. We actually don't really know very much about the differences in tolerance of cold ischemia. I haven't seen any studies comparing males and females there, so they're on even keel until we know more about that. And that's, I'm going to stop there, and we'll talk about the rest of this schematic later on. So in thinking about designing research studies and also in clinical management eventually, perhaps, we do need to think about the implications of donor sex when we're thinking about how to manage donors in both the DCD and the neurological death setting. There may be important factors in terms of organ preservation. So if we can learn more about why or how donor sex has an impact on organ quality, this may influence the way we preserve organs. And then it's also important in understanding graft outcomes, how donor sex plays a role. And this is not to say that we would necessarily select organs based on donor sex. I don't think that that is likely to happen, but it will help us in our understanding of graft outcomes. Okay, so now thinking about recipient sex. So it shouldn't be particularly surprising to anyone that there are significant biologic and physiologic differences by recipient sex. One of which is that there are actually intrinsic differences between XX and XY individuals that may influence the immune response. So the X chromosome has between 1 and 2,000 genes compared with only 50 on the Y chromosome that are implicated in the immune response. So the X chromosome really has the largest number of immune-related genes in the genome. You would think that this would kind of be taken care of by X inactivation. So as you know, one of the two X's in each cell in a female is supposed to be completely inactivated, so only one X is expressed, just like in a male, the one X is expressed. But in fact, X inactivation is not complete. And so somewhere in the range of 23 to 25% of the genes on the inactive X are actually expressed. And this may result in a double dose of some of these immune related genes, which may cause females to have a greater degree of immune inactivation than males. And this hypothesis has been put forward as a potential, or at least one of the potential reasons why girls and women tend to be at higher risk for autoimmune diseases. Clearly, the hormonal milieu differs by recipient sex as well. We just talked about this with respect to donors, but the same is true in recipients. So once the organ is removed from the donor and placed into the recipient, it is now in the recipient sex milieu, regardless of what the sex of the donor was. And so this may have an impact on ischemia reperfusion injury, as well as the hormonal milieu also may influence the immune response and potentially drug metabolism. So this is from a nice review in Nature Reviews Immunology from 2016. I'm not going to read through the entire table, but there have been a number of differences in the immune response, sex differences in the immune response that have been identified. There's differential expression of immune function influencing genes such as FOXP3, IL-2 gamma chain, toll-like receptor 7, and some others, all of which are on the X chromosome. In addition, in general, Estrogen tends to stimulate immune responses. In particular, it stimulates polyclonal B cell activation and inhibits apoptosis of immature B cell promoting antibody class switch, whereas testosterone inhibits IgM and IgG production and enhances apoptosis, inhibiting antibody class switch. So there are some fairly interesting influences of sex hormones on immune function. This is from the same review and nicely shows how differences in immune reactivity between males and females are not constant across the age range. As is well known, the many different things change during development, one of which is the onset of puberty with the development of more sex hormones, and then with aging, the onset of menopause and the diminution of sex hormones, particularly in females. And so this leads to differences in immune activation across the age range. Sex hormones are present on many different immune cell types, and although this is not the case for every single cell, overall, in general, estrogens are known to enhance immune activations, whereas androgens tend to suppress it. We also know that females tend to produce more vigorous cellular and humoral immune reactions than males. And so with the onset of puberty, it's possible that this increase in estrogens may contribute to greater immunologic risk in females compared with males. There are a few sex differences that have also been described in the pharmacokinetics and pharmacodynamics of immunosuppressive medications. But the overall effect is not clearly described. So in terms of calcineurin inhibitors, we know that females tend to have a more rapid clearance of cyclosporin and tacrolimus than males. However, females also tend to have greater intracellular drug levels at the same whole blood level. So it may be that these two different effects balance each other out. They clear more rapidly, but at the same level, they have greater intracellular concentrations. So we don't really know how sex hormones influence this either. It's possible that there may be some pharmacodynamic differences based on sex, influenced by sex hormones, that have not been described. Interestingly, in some of the adherence work we did, we were looking at variability in the trough levels of the immunosuppressive medication tacrolimus, and greater variability is thought to reflect poor adherence, so sort of erratic taking of the drug. However, as I'm going to talk to you about a little bit later, females seem to have greater adherence to medication than males, and yet they also seem to have higher variability in the tacrolimus trough levels. So this is sort of in contrast to what you would expect. If they had better adherence, they should have lower variability. So it does make one wonder whether there may be some other biologic factor that's explaining this higher variability in females than males. Other immunosuppressive medications like mycophenolic acid and azathioprine, we know that these are more rapidly inactivated in males than females, and that the enzymes responsible for this inactivation may be regulated by sex hormones, but there are a few details available about how this really plays out in terms of their effect on the immune suppression. So this plot is the first of a couple. Excuse me. And this comes from a study we did a couple years ago on about 160,000 first deceased donor kidney transplant recipients who were recorded in the SRTR database. That's the US transplant database. And these patients were followed for a median of about 4 years. And what I'm showing here is among recipients of a male donor kidney. We're comparing death-censored graft failure rates at different ages between female recipients and male recipients. So these are unadjusted for other factors. These are just crude, unadjusted rates. You can see that among the youngest individuals, there are substantial differences in graft failure rates, with females having higher graft failure rates than males in these youngest two age categories. This seems to come together here, and in the older two age categories, we're not seeing any differences in crude failure rates. The pattern is a little bit different when the donor is female. Again, we're seeing somewhat higher rates in female than male recipients. In the younger age categories, no big difference here. And now we see a reversal with slightly lower failure rates in females than males in this oldest sort of postmenopausal age category. This plot shows the adjusted and unadjusted differences. So these are hazard ratios for death-censored graft failure, with one being no difference between males and females. This is showing the risks in females compared with males. So in the first case, these are all recipients of a male donor, and these are the different age categories. And I should mention that these were age-dependent categories. So patients would get their transplant at whatever age they got it, say 15, which would put them here to start. And as they were followed over time, if they were followed long enough, they would go in, their observation would go into the next age category, and so on. So we'll focus just on the adjusted hazard ratios, and these were adjusted for all other potentially confounding factors. So the actual numbers are down below of the adjusted. So you can see that across all age categories, female recipients had a significantly higher risk of graft failure than male recipients, although the magnitude of the effect decreased with increasing recipient age. So the highest hazard ratio was about 50% higher risk in the youngest age category, down to only 5% higher risk in the oldest age category. That was when the donor was male. When the donor was female, the pattern was quite different. So here we're seeing no material or statistically significant difference between female and male recipients in the youngest age category, but females still had a significantly higher risk, 28% higher risk of graft failure than males in this adolescent young adult age category, which then was still slightly higher, but not statistically significantly higher in the middle adult age category. And in the oldest age category, it actually reversed. So females had a significantly lower risk, not a big difference, only 5%, but a significantly lower risk of graft failure than males. So before going on to kind of try to explain why we saw what we saw in these two plots, I think it's important to consider how gender might play a role as well. Now, in the study we didn't measure gender specifically, so we used sex as a surrogate for gender. Gender, as I'm sure you're aware, is the socially constructed roles, behaviors, expressions, and identities. So we're not talking about biology here. We're really talking about what are the socially constructed factors. And it's important to recognize that gender may also have an influence on graft outcomes. I'm going to focus mainly on the influence it may have on adherence to immunosuppressive medications. So adherence is known to be a very, very important determinant of long-term graft outcomes. And clearly gender differences in beliefs, values, or behaviors that influence medication adherence may also influence graft outcomes. Gender may also play a role in access to transplant. So we know that there's poor access to both living donor and deceased donor transplant for women than men, and gender probably plays a role in at least some of this, also in donation rates. These are things that Dr. Melk had talked about in some detail in her webinar. And so if you did not get a chance to hear that webinar and are interested in that, I'll refer you to her talk where these are covered in more detail. These are data from the Take It study, which was a randomized trial of an intervention to improve medication adherence in adolescent and young adult kidney transplant recipients. This is a secondary analysis of the data from the Take It study. Here we're comparing medication adherence as measured using electronic medication monitors in female versus male participants. The data that I'm showing here were from the 3-month run-in period. So this was before the intervention actually started. We stratified the patients by age, with younger patients on the left here and older patients between 17 and 24 years of age on the right. Females are the little black circles and males are the open triangles. And it's pretty easy to see that in the younger individuals, there was no clear difference in adherence between males and females, whereas in the older age categories, females tend to have substantially higher adherence than males. This table just shows the adjusted analyses. So this is the results of an ordinal logistic regression. Taking adherence was just if they actually took their medications, didn't matter whether they took them on time, and timing adherence was taking their medications within 2 hours of when they were meant to be taking them. And regardless of what we adjusted for in our models, we were seeing a sort of 2.5 to 3 times higher likelihood of good adherence in females compared to males for both taking and timing. Whereas that was only in the older age category, whereas in the younger age interval, there were no differences between males and females in adherence. And this is not particularly surprising since in the younger kids, their parents are primarily responsible for it, whereas in the older age group, this is when they're taking responsibility themselves. Okay. So this table just tries to put together all the potential factors that we could think of that might influence graft outcomes and may differ by sex across different ages. So in the top part of the table, we're thinking about when the donor is male, and in the bottom part when the donor is female. And these are the different recipient age categories because each of these factors may differ in different age categories. So just taking it one age category at a time, in the youngest individuals, we don't really expect sex hormones to be playing a big role since most of these patients are pre-pubertal. Now you could argue that many of those in the sort of 12 to 14-year-old age category were post-pubertal, and that would be a reasonable argument. We actually did a sensitivity analysis among those under the age of 10 and got the same results. So no impact of sex hormones. When the donor is male, you would expect that potentially a female recipient may have an immunologic reaction against the HY antigen, increasing her risk of graft failure compared to a male recipient who would not have that reaction. In terms of adherence, as I just showed you, there's no significant difference in adherence between males and females in this age group, so we wouldn't expect it to play much of a role. And then there may be intrinsic differences in the immune system based on gene expression on the X chromosome that would contribute regardless of the age of the individual. So if we sort of add up these combined effects across all ages, we would expect a higher risk in females compared with males. In the adolescent young adult age group, now sex hormones would be playing a larger role. HY antigen would be unaffected. It would be same thing regardless of the age category. In this age group, we'd start to see better adherence in females than males, so that may mitigate some of the higher risk that one would expect based on the influence of sex hormones, the HY antigen, or sex chromosome effects. We'd expect similar things to be playing roles in this age category. Perhaps the effect of sex hormones may be somewhat diminished with increasing age. I'm not really sure. And then once you get to those over 45 years of age, so in the sort of postmenopausal category, the role of sex hormones would be much diminished here. And the other factors would be similar to other age groups. The differences in gene expression on sex hormones, again, it's not clear what impact that might have. It may actually be a little up arrow, or it may be diminished in this age group. In the lower part of the graph, this is in the setting of a female donor, and all the factors in this table are the same as in the table above, except for the HY antigen, which clearly won't be playing a role when you have a female donor. So taking all these together, the contrasting and different direction impacts may help explain the different patterns we saw across different ages for female and male donors. We're coming back to the same schematic that I showed you at the very beginning of the talk. So this little dotted line is when the organ is removed from the donor and goes into the new environment of the recipient, where we talked about estrogen potentially protecting the recipient from ischemia reperfusion injury and testosterone augmenting this injury. This may have an impact on delayed graft function. And these are all the factors that we just discussed that may play a role in long-term graft function. And if you just look at the number of little female symbols and the number of male symbols, there's a preponderance of them in the worst graft survival section of the schematic, and a preponderance of male symbols in the better graft survival section. So these are all hypothesized. There is some evidence both within transplant and outside transplant to explain some of these, but more data is needed before we can really say exactly what's happening. So we're getting close to the end. To summarize, I guess I'd like you to think about when you're designing your own research studies or when you're reading studies in transplantation, it's important to consider these potential influences of sex and gender. In basic science studies, we need to pay attention to the sex of the cells and the animals that we're using. In studies of donor management, in studies where we're considering better ways to do organ preservation, and we really do need studies looking at the pharmacokinetic and pharmacodynamic effects of sex hormones, and just of whether these things differ by sex, regardless of whether sex hormones are the mediator. In clinical science, we need to pay attention to the donor sex and the recipient sex, as well as the recipient gender. All of these things may have an impact on access to the wait list, on access to transplantation, and Dr. Melk spoke about those factors more in her talk. They may also influence graft outcomes and patient outcomes. And another thing to consider is that behavioral interventions that we may be developing may actually need to be gender specific. So one gender may respond in different ways to a behavioral intervention than another gender, just like one sex may respond in a different way to a drug intervention than the other sex. This approach to science is relatively new, and so we're only just starting to learn more about the impact of sex and gender on transplantation. At very least, when we're doing analyses, we should adjust for sex as a confounder, but usually that is not sufficient. It's very important both in large epidemiologic studies, but also in basic science studies, to try when possible to actually do the experiments or do the analyses stratified on sex. That will allow you to determine whether the impact of whatever it is you're looking at actually differs in males than it does in females. If you can't stratify on sex, then at least report the sex of the cells or the animals that you used. If we don't consider the impact of sex or gender on outcomes or in our experiments, it may actually lead to erroneous conclusions. I think we've covered a lot of these points already. Yeah, we have. The goal in the end of considering sex and gender in our research is to improve outcomes by allowing us to develop sex-specific strategies. That might be sex-specific strategies to immunosuppressive medications, but it may also be sex-specific strategies to organ preservation or to donor management, the goal of which is to improve outcomes for individuals of all ages and both sexes and all genders. I'm going to end just thanking my collaborators, and I guess we can open it up for questions now.